I'm so pleased today to introduce Koa Matsuoka. Koa was born in Honolulu, and he was an undergrad at UH Manoa in the Natural Resources and Environmental Management Department. Koa, I asked him yesterday what motivates him, and Koa said that he's motivated by ensuring that future generations have the same access to the natural and cultural resources that he has today. He's interested in facilitating, as he says, good change without the loss of things that make Hawaii unique. So I think you'll see that spirit represented in what Koa is talking about today for his master's thesis. I first met Koa in the summer of 2011 when he worked as a pipes intern. It was funded through the EPSCoR project. He worked with myself and a postdoc on the project, Bruce Dudley, and he worked on the ecophysiology of kiawe trees. Koa distinguished himself as hardworking, diligent, and conscientious, and I went back to the letters of reference that we wrote at the time to get those terms. In 2017, Koa came to me about wanting to start a master's project, and he wanted to work on seed dispersal, and I was really pleased to welcome Koa back into the lab group. Over the time that I've known him, he's matured greatly as a scientist and as a leader. He's one of our Native Hawaiian emerging professionals that are managing Hawaii's natural resources, and I'm really excited about his current position that he's been working on for several months as it's a PCSU position working with the Division of Forestry and Wildlife Office in Honolulu as a Protected Species Habitat Conservation Planning Associate. He's also worked projects with the San Diego Zoo Global. I also want to mention that Koa is an excellent nature photographer. You will see pictures and the background that he's using today that he's taken himself. And you can follow him on Instagram at Koa Mats, M-A-T-Z. So I'm really proud of Koa, and I'm excited for you to all hear and see his work today. And so I pass the bar off to Koa and take it away.
All right, mahalo Becky for the introduction. Aloha mai kako. Today I'll be discussing my master's research that examines seed dispersal and germination by native versus exotic avian frugivores on Hawaiʻi Island. Pictured here are the 4 avian frugivores included in my study. So before I start, here is a brief overview of what I'm going to talk about today. First, I'll go over what an avian seed disperser is, or what avian seed dispersal is, and its importance in Hawaii, followed by a brief introduction of each of my study species. Then I'll go over the methods used to collect fecal, seed rain, and germination.
Whatever.
Lastly, I'll discuss what the results mean for forest health and how this information can be applied to real-world management strategies. So first off, what is a frugivore? A frugivore is an animal that subsists totally or primarily on fruit. This includes birds, monkeys, bats, and tortoises, to name a few. So why is avian seed dispersal so important to ecosystem function on tropical islands? Well, as a result of thousands of years of coevolution and isolation, birds and plants formed mutual relationships where plants provide sustenance with fruit, and birds consume that fruit and spread the plant's progeny. In Hawaii, this relationship is evident as a majority of fruiting plant species were exclusively dispersed by native birds due to the lack of terrestrial mammals. Many plant species develop fleshy fruits to appeal to birds and promote consumption. The lobeliads of Hawaii are a prime example of this. However, since the arrival of humans, over half of Hawaii's endemic avifauna have gone extinct. As a result, ecological processes such as seed dispersal have been disrupted due to wide-scale extinctions. Many remaining fruiting plants now lack their primary disperser and are limited to small remote areas of protected habitat. So why do native frugivores matter? Well, in general, the avian community benefits fruiting plant reproduction by increasing germination rates and recruitment success through gut passage and dispersal away from conspecifics. These benefits are essential for facilitating primary succession and promoting the natural regeneration of fruiting plants in the natural landscape, especially after disturbance events. Only in the last century or so, exotic frugivores have been introduced to Hawaii and spread quickly across the archipelago. The birds that fill the empty niches left by extinctions of natives are usually smaller in size and have generalist diets consisting of a wider variety of food resources. One such exotic frugivore is the warbling white-eye. Formerly known as a Japanese white-eye, the warbling white-eye is the most common bird that represents this trend of supplanting native frugivores. And for the rest of this presentation, I will refer to the warbling white-eye by its four-letter code Wawei. Wawei were brought to Hawaii during the plantation era from Japan. They were initially introduced to Oahu, then eventually spread throughout the archipelago. According to the Hilo Tribune Herald, in 1937, 252 birds were introduced to Hawai'i Island to help eradicate insects in the ag fields. On Hawai'i Island, this bird is wide-ranging from very dry to wet habitats from sea level to tree line. The white-eye family has been successful at colonizing oceanic islands more than any other avian family, and they are opportunistic generalists whose diet consists of fruit, nectar, and insects, and they thrive in disturbed environments and island ecosystems. The Wawei family is found in tropical areas from Africa, Asia, and throughout Oceania. I need a sip of water. The other exotic species I looked at was a red-billed Leiothrix. And for the rest of this presentation, I'll refer to the Leiothrix by its four-letter code, Ribley. This bird was initially brought to Hawaii in 1911, and similar to the Wawei, the Leiothrix is found in a range of habitats from sea level to subalpine forests.
I just want to remind everyone to please be on mute.
Is there a rock button? Okay, thanks. So similar to the Wawei, the Leiothrix is found in a range of habitats from sea level to subalpine forests. It can be found in both native and exotic dominated Hawaiian forests. The Leiothrix is considered a generalist and spends the majority of its time in the forest understory foraging on fruits and arthropods. The Leiothrix's generalist diet and resilience to avian diseases allows it to thrive in a variety of different habitats, leading to its wide distribution. The first native frugivore I looked at was the ʻalalā, or Hawaiian crow. This bird is endemic to Hawai'i Island, and its historic range once encompassed the moku of Ka'u and Kona on the leeward sides of the island. The ʻalalā is currently listed as critically endangered and was formerly noted as extinct in the wild in 2002, until releases back into the wild started in 2016. There are currently about 120 plus birds in conservation breeding centers derived from 9 original founders. There are also released individuals remaining at the Pu'u Maka'ala Natural Area Reserve release site on Hawai'i Island. Historically, the ʻalalā consumed insects, bird eggs, nestlings, carrion, and up to 30 species of Hawaiian fruiting plants. In Hawaiian culture, the ʻalalā was regarded as an aumakua, or family guardian to some. And the ʻalalā was also known as an animal that led spirits to a lele, or jumping site where spirits leapt into the afterlife. The word ʻalalā refers to the cry of a baby or a loud voice, which represents its loud call very well. ʻalalā is also a style of oli or Hawaiian chant, and this bird is referenced in a number of Hawaiian songs and chants for its call. During warfare, the ʻalalā chanters were the ones who called out the commands of chiefs during battle and initiated Hawaiian ceremony at heiau, or religious sites. The second native frugivore I looked at was ʻōmaʻo, the Hawaiian thrush, or Myadestes obscurus. The ʻōmaʻo is endemic to Hawai'i Island and listed as vulnerable, but considered stable in areas where it is found on the windward side of the island. However, the ʻōmaʻo has been absent from the leeward side for decades now. The ʻōmaʻo is considered the last functionally extant native frugivore in Hawaii. This means it exists in high enough numbers to provide landscape-level ecosystem services. The other extant member of the Hawaiian thrush family is the puaiohi, which is endemic to Kauai, but its population is around 500, and it's listed as critically endangered. The ʻōmaʻo's diet consists mainly of fruits and insects. Thrush species, including the ʻōmaʻo, were once broadly referred to as ʻaʻmaui, meaning bird of Maui, alluding to the shape-shifting demigod mentioned throughout Polynesian legends and Moana. Due to this reference and stories mentioning Maui, had other bird forms besides the hawk and the white rooster, there was speculation Hawaiian thrushes could be a forgotten kinolau or body form of Maui. Although ʻōmaʻo usually means green in Olelo, Hawaii, and this bird is gray, the word ʻōmaʻo can also mean to nag or beg like a child. Given the ʻōmaʻo's tendency to tremble and its quivering, croaking-like call, in Old Hawaii, the name ʻōmaʻo was derived from the description of the bird that would beg for warmth. And mahalo to Noah Gomes for this insight. So anyway, here's a map showing the former and current ranges of ʻōmaʻo, or range of ʻōmaʻo, and my 2 study sites, which includes the release site of the ʻalalā. My study sites are the Power Line Road Kipuka, marked by the red star, and the Pu'u Maka'ala Natural Air Reserve, marked by the yellow star. As you can see, the pink area represents the range of ʻōmaʻo that once extended on both sides of the island. Keep in mind, the current range is encompassed within the former range. Today, the ʻōmaʻo are mainly on the windward side of Hawaiʻi Island. This dramatic decline is attributed to the introduction of avian diseases such as avian malaria and pox, invasive predators such as rats, cats, and mongoose, and loss of understory fruiting plant habitat caused by feral ungulates such as cattle, pigs, and sheep. Similarly, the ʻalalā range map shows the same trend. The origin of the ʻalalā is unknown, but researchers speculate they came from the Australasian region. There are 2 other extinct species of Hawaiian corvids that once existed on Oahu, Molokai, and Maui. These species were easy to distinguish from ʻalalā as they had a larger body size and different shaped bill, as you can see in the figure here. Although there is possibility ʻalalā existed on Maui, Banco and Banco 1980 noted the historical range of the extant ʻalalā, Corvus hawaiiensis, was limited to Hawaiʻi Island even before a sharp decline in the early 1900s. This is shown in the V-shaped belt here, consisting of woodlands of dry woodlands of ohia and koa forests from 500 to 1800 meters in elevation. Similar to all the other native birds, the decline of the ʻalalā is attributed to diseases such as avian malaria and pox, but also toxoplasmosis, habitat loss from land use change, such as ranching, and the introduction of invasive predators, again, rats, cats, and mongoose. As mentioned earlier, the ʻalalā now only exists in conservation breeding centers and at the release site at the Pu'u Maka'ala, NAR. So to examine seed dispersal differences among native and non-native frugivores, the Pu'u Maka'ala Natural Air Reserve, or NAR, was one of 2 release sites. NARs are parcels of land designated by the state of Hawaii as the best remaining examples of Hawaiian ecosystems and are given the highest level of protection and restoration. This particular NAR exists between 1,600 and 1,800 meters above sea level and consists of native dominated forests protected by fences from ungulates. There's also ongoing predator control for rats, cats, and mongoose. This is also the site where ʻalalā released in cohorts, where ʻalalā were released in cohorts starting from 2016 to 2020, or 2019. During peak survival, there were up to 27 ʻalalā inhabiting Pu'u Maka'ala from 3 different cohorts. My second study site was the Kipuka system off Power Line Road, which is located in the saddle between Mauna Kea and Mauna Loa. The site provides habitat for a variety of native and introduced bird species and understory fruiting plants. The Kipuka system is part of the Upper Waiakea Forest Reserve and exists between 1,500 and 1,700 meters above sea level. It consists of native-dominated montane forests naturally fragmented by lava flows. The remaining forest islands are referred to as Kipuka. This site is not fenced from fairy ungulates and does not have active predator control. So here are the common fruiting plants that dominate the understory of the upper montane forests in my study sites. This table shows the 9 fruiting plant species that were found in the diets of the avian frugivores in my study. I lumped the plants whose seeds were difficult to identify among species as just a broader genus. For example, species within the genus Vaccinium and Coprasma, Ohelo and Hilo, are lumped together in the table here. The table also shows the average number of seeds found per fruit of each species or genus. From now on, I'll refer to these plants by their 6-letter codes and Hawaiian names. Here's another table showing the seed sizes of the different plant species. Note the range in size among these species, and remember the small-seeded plants highlighted in red, and the medium-sized seed highlighted in orange. These small-seeded plants consist of rubhal, akala, vaccinium or ohelo, and ileano or kava'u. And the medium-sized seed was olapa or shed tree, Chirodendron trigen. So the first question in my study was, what native plants will be dispersal limited in the absence of native frugivores? I attempted to answer these questions by measuring avian seed rain and fruit preference through diet composition. I hypothesized that smaller seeds would be more prevalent than larger seeds in seed rain traps, and that native frugivore diet diversity would be higher than exotic birds. So to do this, first to measure avian seed rain, I placed hanging mesh baskets 1 meter in diameter above the fruiting plant understory. In theory, these baskets exclusively catch avian dispersed seeds because they hang from dominant canopy trees that do not use birds for dispersal, such as koa and ohia. From this, I can deduce that any seeds identified as fruiting plants in the baskets below got there from being eaten by a bird, then pooped into the basket from a bird perching above it. Now, you would think that the odds are pretty low for a bird to poop in such a small area, but I did find a good amount of seeds, especially from traps in the interior of the kipuka. So in total, 48 of these baskets were randomly distributed among 4 kipuka fragments and left out to collect seeds for 8 months. For logistical reasons, I did not sample seed rain at Pu'u Maka'ala nar. And every 2 weeks, seeds were collected from the baskets and identified using a seed reference library. To measure fruit preference or diet composition, we misnetted birds in the kipuka system with Christina Paxton in the Lohe lab. I also volunteered with the NAR's banding crew, Alex Wong, in the Pu'u Maka'ala nar. So birds were caught in mist nets, then extracted and placed in bags if they had not already pooped in the holding bags. The fecal was then placed in vials and labeled appropriately. In addition to collecting poop, a lot of other data was collected for other research projects going on in the Lohe lab and with NARS. ʻAlalā fecal samples were collected incidentally from supplemental feeders by the ʻalalā tracking team. And during this collection period, no Hawaiian fruiting plants were provided in the ʻalalā supplemental diet. These poop vials were then taken to the lab and seeds were sorted from the fecal matter. These seeds were then counted and identified to the species or genus level using a seed reference library. Pictured in the middle, this photo in the middle shows Pukiave, Olapa, and Kukainene seeds, and the photos on the right are the seeds from the reference library I used to match and identify these seeds. So moving on to the results. From this pie chart, note that the small-seeded vaccinium or ohelo is represented by the blue color. You can see that ohelo made up a little more than half of all the seeds found in the seed rain baskets. Then Ilex anomala or kava'u, another small-seeded species, made up the next highest proportion. And Chirodendron triginum or olapa made up the highest proportion after that. Moving on to the diet samples, the first metric here is species richness in the diet. The y-axis shows the mean number of species found per fecal sample, and the x-axis shows the avian frugivores. ʻōmaʻo stood out with the highest species richness, followed by ʻalalā and Ridley, and wawe with the lowest species richness. The second metric here is seed abundance in diet. The y-axis shows the mean number of seeds found per fecal sample, and the x-axis is the avian frugivores that consume them. Again, ʻōmaʻo stood out with the highest species or seed abundance, followed by ʻalalā and Ridley, and wawe with the lowest seed abundance. The last metric that I was most interested in was Shannon diversity, which considers species richness and evenness from seed abundance. The y-axis shows the mean Shannon diversity per fecal sample, and the x-axis are the avian frugivores. Again, ʻōmaʻo stood out with the highest diversity, followed by ʻalalā and Ridley, which is not significantly different from each other, and Huawei with the lowest diversity of all. So when we look at the diet composition, you can see the small-seeded plants, shown in the pie charts by the blues and purples, made up the majority of the plant seeds found in the frugivores diet. These pie charts show the relative abundance of fruiting plant seeds in the 4 avian species by seed. Relative abundance is calculated as the total number of seed from a particular plant species divided by the total number of seeds collected from the fecal of the avian frugivore it was collected from. You can see small-seeded plants such as vaccinium, orohelo, and rupal akala make up the majority of the seeds found in all 4 avian frugivores. Also note that Huawei only consumed 3 small-seeded plant species, and vaccinium made up the majority of its fruit diet. So in this graph, we have seed abundance, but I converted it into fruit. Some species have many seeds in their fruit, like ohelo, while some have fewer but larger seeds in their fruit, like kolea or merles. This was calculated by using the known average number of seeds found in the fruit of a particular species divided by the total number of seeds. For example, on average, olapa has 3 seeds. If I found 12 seeds in the fecal sample, that would mean the bird likely consumed 4 olapa fruit for that fecal sample. The pie chart shows a different diet composition because ohelo have around 250 seeds per fruit, whereas merles or kolea only has 1. This disparity in number reflects the dispersal strategies of each fruiting plant species, as there are differences in seed viability. Okay. This pie chart shows the relative frequency of fruiting plant seeds that comprise the overall diet of each avian frugivore. Relative frequency is calculated as the number of fecal samples containing an individual plant species divided by the total number of fecal samples collected from each avian frugivore. The brown color shows the proportion of fecal samples for each frugivore that did not have any seeds in them. Note that the exotic frugivores like Ridley, and especially the Huawei, have the highest proportion of fecal samples with no seeds. In contrast, ʻōmaʻo and ʻalalā have fruiting plant seeds in more than 75% of all the fecal samples collected. This is even while released ʻalalā are being supplementally fed. Another important finding was there were no non-native fruiting plants identified in any of the fecal samples. And this was not surprising because although there are some exotic fruits like thimbleberry at the study sites, they're relatively rare and fruit seasonally. So circling back to the hypotheses. For hypothesis 1, the seed rain baskets had a much higher abundance of smaller seeds such as vaccinium, ohelo, than larger seeds like mersani or kolea. So that checks out. For hypothesis 2, the diet diversity of native birds, especially ʻōmaʻo, were generally higher than that of exotic birds like Huawei. However, as stated earlier, ʻalalā and Leiothrix were not significantly different in their diet diversity. But overall, native frugivores did have higher diet diversity than exotics. So you can check that off too. Okay, moving on. My last research question is, how do exotic frugivores compare to natives in facilitating plant recruitment? To answer this question, I compared gut passage effects on germination success between the 4 frugivore species. For this, I hypothesized that the evolutionarily adapted digestive tract of native frugivores will scarify seeds and remove pulp more effectively, therefore yielding higher germination success relative to corresponding controls. The seeds with no gut, therefore yielding higher germination success relative to corresponding controls than seeds with no gut passage and seeds passed through exotic frugivores. So to measure germination, I took the same seeds from the diet samples and placed them in growth media and germination trays. The germination trays were placed in a greenhouse at Keauhou with conditions relatively similar to the conditions at the study sites. All seeds were watered regularly with timed irrigation and placed in the same 3:1 coarse sand to vermiculite ratio. I made controls that had seeds with and without the pulp attached to determine if gut passage was actually the variable affecting germination. And to make the controls without pulp, I just manually remove them by hand. So in the end, there were mixed results with the germination trials. I did not have enough germinated seeds from exotic frugivores to compare to the natives. So I compared gut passage treatments through native frugivores relative to the controls. These line graphs show germination over time among 5 fruiting plants that I had large enough sample size to actually look at. The y-axis is a percentage of total seeds germinated over time since seeds were sold on the x-axis. The colored lines represent a different control or gut passage treatment through a particular frugivore. The notable red line represents the control with the pulp still attached as a baseline comparison of gut passage effects on germination rates. You can see there is variation in germination rate relative to controls with pulp still attached, but overall, in all 5 plant treatments, overall, treatments in all 5 plant species had faster initial germination than controls with pulp still attached. But this trend plateaued as time progressed. When you look at the total percentage of seeds that germinated under each treatment, you see that vaccinium had the most significant difference between gut passage through birds and controls with and without pulp. In these bar plots, the y-axis shows the percentage of seeds that germinated and the x-axis is the treatment type. The letters mark significant differences in germination percentage between the treatment types. From the figure, you can see larger seeds such as Merles or Colea and Coprasma Hilo did not have any significant differences between the proportion of seeds that germinated, while Shetri, Olapa, and Cockburn Kukainene had some varied results. So coming back to my final hypothesis, I was not able to determine if seeds that underwent gut passage through native frugivores had more germination success than exotics due to sample size limitations. Unfortunately, I did not acquire enough seeds that went through exotic frugivores and germinated to make a comparison between them. So that didn't come through. However, I did find that controls with pulp still attached had lower germination success than seeds with no pulp. This includes both controls with pulp manually removed and seeds that went through the gut of ʻōmaʻo and ʻalalā. Okay. So in conclusion, well, I got plenty of time. Exotic passerine frugivores included in the study are partial substitutes for native frugivores. While exotic birds, especially Ridley, do disperse native seeds, native birds are more frugivorous than exotics and disperse a higher diversity of fruiting plant species. Therefore, fruiting plants with larger seeds such as Colea and Nile are less likely to be dispersed if ʻōmaʻo and ʻalalā go extinct. Now, this is somewhat consistent with a study by Foster and Robinson 2007 that looked at seed dispersal in montane forests of Maka'ala, Maui, where all native frugivores are extinct. They found that fruiting plant community composition reflected the plants commonly found in the diet of the exotic frugivores that inhabited the area. This trend suggests that birds directly influence the composition of plant communities in Hawaii. Now, seeing how the majority of the plants represented in diet samples and seed rain were from smaller seeded plants, this trend may exclude many larger seeded plants from persisting in the future without human intervention. And we already see this trend with the decline of some larger seeded plants like halapepe and hoava, which are known to be dispersed by ʻalalā when the spread was abundant. Another key result was that pulp removal was generally more important than gut passage in influencing germination rate and percentage. And this is especially true for smaller seeded plants like ohelo. So when we apply this concept to restoration, removing pulp manually to expedite germination could serve as an important management tool. Anecdotally, native plants generally germinate slower than invasive plants. Therefore, removing pulp and giving native seeds a competitive advantage at colonizing restoration sites may serve as an important step to initiating forest regeneration. These results also give us initial insight into the role of ʻalalā as an important seed disperser in the wild. Given that over 75% of ʻalalā fecal samples already contain fruit, even while being provided daily supplemental food and being in the wild for 1 to 2 years, these birds seem to have retained their innate foraging ability after generations and decades at breeding centers. And they seem to be adjusting well to the wild as they're utilizing novel food resources available to them. Another interesting result was that a good proportion, about 20% of ʻalalā fecal samples, had kukainene, or copper and seeds in them. This plant was not fed to birds at breeding centers and grows on the ground in open forest patches. This suggests that ʻalalā are consuming novel fruit and finding it in places where they are vulnerable to eel predation. I did not find ʻōhawaii or Clermontia and hoʻawa in any fecal samples of the ʻalalā. And these are plants that historically relied on ʻalalā for dispersal and are available at the release site. Another interesting observation is that released ʻalalā were observed eating maile fruit, which has seeds almost double the size of the largest seed in this study, which was kolea. While released ʻalalā haven't completely weaned off of supplemental feeders, they are showing positive signs of moving in that direction. In the future, when all threats to ʻalalā can be mitigated for, and we successfully figure out how to establish ʻalalā in the wild without human intervention, future applications could include using reintroductions of native frugivores as a means to restore seed dispersal function in places where they are extinct, which is pretty much every other island except Hawaiʻi Island and Kauai. Because other Hawaiian corvid and thrush species were historically found on other islands, reintroductions could more easily be applied to neighbor islands, as those niches are empty. Ultimately, these results will help us assess forest resilience to disturbance, regeneration ability, and give us insight into how plant community composition will change over time. So as is common with most experiences, there were a lot of issues that came up. There were logistical complications in monitoring seed rain traps, and Hurricane Lane came and knocked down more than half of the aerial seed rain traps I set up. These traps were mostly in exposed areas in the forest edge and in the Kipuka matrix. These events changed my original goal of comparing seed rain composition at varying distances in and around Kipuka to just looking at total seed rain composition in the Kipuka. For germination trials, there were limitations due to the amount of time it took for seeds to germinate, which is usually between 2 to 10 months, and the logistics of monitoring the germination trays consistently enough to get a narrowed down time frame of when seeds actually germinated. Ultimately, this high variation in seed germination data made it unfeasible to run a generalized linear model as originally planned. So looking forward, future research directions should attempt to obtain a more robust and even sample size that have more power to detect notable differences in germination success between gut passage treatment types. It would also be interesting to compare avian seed rain at sites with and without ʻalalā to look at their impacts on landscape level seed dispersal. Another interesting topic would be to monitor the ʻalalā diet composition over time and look how diet shifts pre and post weaning off of supplemental feeders. Additionally, one component of seed dispersal I didn't find in the literature was how Ridley foraging patterns compare to ʻōmaʻo and ʻalalā. This would provide insight into how their movement how Ridley movements influence seed dispersal distance and deposition into locations ideal for germination and survival. Moving ahead, there is good news at the Konahema Preserve, where the Nature Conservancy purchased a parcel of land on the southern leeward side of Hawaiʻi Island. This is the same site where the last group of ʻalalā existed until going extinct in 2002, and there were ongoing talks among state and federal agencies of potentially using this site as a new ʻalalā release site. The Konahema Preserve also has ʻōmaʻo slowly moving in from Ka'u and establishing within its borders, which is one of the first signs of them coming back to the leeward side of the island in decades. This scenario provides a good opportunity to look at pre and post impacts of reintroducing native frugivores back into the landscape where they have been absent for decades. So with that, I'd like to give a big mahalo To all these people, organizations, and friends who helped me collect data, provided guidance, and overall made this a great experience. Mahalo to my committee, Hilo, Alex Wong, and Brett Mossman from NARS for collecting fecal samples, Becky Ostertag, Bryce Masuda, and Christina Paxton for mentoring me, Doreen Koizumi for all the help with finances, Jasmine Curio from Pipes and all the people who helped collect seed rain. A big mahalo goes out to Ariel Imoto, who monitored the germination trays while I was working full-time, and Kinori for pushing me during quarantine to finish this thing. Also, mahalo to the Hau'oli Maoloa Foundation for giving me the opportunity to travel to conferences. This wouldn't have been possible without your support. Now I can focus on my job. So with that, I will take any questions. Mahalo.
Thank you so much, Koa. And yes, we're open for any questions.
Hi, Koa. Can you hear me? This is Paul Banco.
Yeah, I can hear you.
Oh, great presentation. Thanks very much for that. Yeah, I'm just curious, and obviously this wasn't part of your study, but maybe you have an idea about it since you've been looking at all these fruits and seeds. Do you think that insects that will be inside of some of the fruits, do you think that would influence the foraging strategy of these frugivores? That is, do you think they might be, in some instances, selecting fruits because of potentially a higher incidence of insects inside the fruit?
Yeah, that's definitely a possibility. I think the birds would probably get two benefits from foraging on fruits that have insects and fruit meat. So yeah, that's definitely a possibility. Sorry, I'm blanking right now.
No, that's fine. I mean, it wasn't part of your study, so I don't expect you to know, but I just curious if in opening fruits and extracting seeds and that kind of thing, if for example, maybe Ohelo, I'm thinking might be a candidate fruit that birds might target,
yeah.
partly because of insects.
Yeah, when making the controls, many of the fruits didn't have any insects in them. And then when I dissected the fecal samples, for the native frugivores, a lot of them were either had a higher proportion, like a lot of seeds in them, or a lot of insects. Whereas a majority of the exotic frugivore fecal samples had majority insects and some seeds in them. So
yeah.
Yeah.
Yeah, the reason it comes to my mind is partly because of the behavior of the kiwikiu, the Maui parrotbill, which will kind of scissor off the tops of Kanavau, I think, and extract insects from that without eating the fruit actually, but just sort of extracting the insects from there. One other question, if I could, is what would you say is the likelihood that smaller seeds may be destroyed in the gut at a higher rate than the larger seeds? I mean, is there evidence of very much actual seed destruction in the gut of the bird?
Yeah, so in going through the fecal samples, I didn't really find much evidence of that. From what I can remember, almost all the seeds I found were intact. Even the larger seeds. So yeah, just in going through the samples, I didn't really find much seed destruction.
Yeah, so
Even
the evidence is mostly that they're dispersing seeds and not destroying them.
Right.
Yeah, cool. Thank you.
Yeah.
Other questions?
Can I go?
Sure, Nikolai, and then whoever, I missed who that was, but you're next.
It's not really a question, it's more of a comment. I'm actually trying to grow Vaccinium right now from seed, and it was interesting for me to see the difference. It's had a practical application.
Yeah, there's definitely a huge noticeable difference when you just take them out of the pulp, but not really much difference after after the seed's going through the gut. So yeah.
Oh, I thought in the Vicinium that it had a huge difference going through the gut, and that going through the pulp, removing the pulp didn't provide as much of a positive effect. Maybe I saw that wrong.
Yeah, just overall, the seeds being outside of the pulp, they germinated a lot faster. It's because they're all compacted in the fruit. So for the seed coat to be penetrated and water and air to get in there to initiate germination, it's easier when they don't have to go through the fruit pulp layer. So yeah.
Thanks.
My turn?
Sure.
Cool. So Koa, I guess my question, maybe I missed it when you addressed this in your last slide about future ideas, but do you think the beak size has anything to do with the abundance of fruit species found in the birds' diets? You know, ʻalalā and ʻōmaʻo are larger birds than ribleys and wawe. So do you think maybe the next idea would be to look at generalist game birds like quails and Kalish pheasant and maybe Urkel Franklin?
Oh yeah, I put that in my slide, but I forgot to say it. So yeah, huame, or the melodious laughing thrush, which is a relative of the ʻōmaʻo, also exists in these forests, but I wasn't able to put that in my study because they're so rare, or they're rare in the study sites and they're harder to catch. But yeah, that's definitely another frugivore that should be looked at, in addition to the Kalish pheasants, which are galliforms that are larger birds with a larger gape size that in theory could consume a wider variety of fruits. And I know that this is actually being looked at Hakalau with USGS, so can stand by for the results on that. But yeah, I don't know if I mentioned it, but I think gape size has everything to do with the range of fruits that a bird is able to consume, which is the reason why we see larger exotic frugivores, ʻōmaʻo and ʻalalā, at a higher diversity. So yeah.
Thank you, Koa. So Becky, just to let you know, somebody asked a question in the Zoom group chat.
Okay, so from Chuck Camara, nice study. Did you observe any evidence of rodent seed predation, i.e. dropping seed fragments in seed traps, that could have affected the seed rain data?
No, I did not find any damaged or rodent predated seeds in the seed rain traps. Pretty much, yeah, all the seeds I found were intact and good.
I have a question about the ohelo seeds. I think it was in the seed traps. You said that these were the most abundant. Is it possible that they could have dispersed there by themselves without bird influence?
No. So I mean, the only way would be they'd have to defy gravity, because I hung the seed rain traps above all the ohelo plants. So these seed rain traps were anywhere. I made sure that they were the only plants above them were ohia and koa. So in order for a ohelo seed to get there, one of the few ways would be consumption by a bird and then being pooped in the basket, or like the last question, a rat could have taken the fruit and then dropped it or pooped it into the basket. So yeah.
Other questions?
Yeah, this is Chuck Camara again. Can I ask one more question?
Sure.
Yeah, that was a really great study. I can appreciate the difficulty of getting that kind of data in the field. I was actually pretty amazed that you did not find any non-native seeds in your seed traps. And you had mentioned that thimbleberry was one of the fleshy fruited non-natives in your study site. And I was wondering if you thought that the lack of the non-native seeds was due to the general absence of fleshy fruited non-native species at your site, or that the frugivores you were looking at were preferentially going for native fruits? Like, were there any other non-native fleshy fruited species or avian dispersed potential plants that were not being dispersed by them despite their presence at your site?
Yeah, so there are other studies that looked at that in more invaded degraded forests, and they did find that exotic frugivores do disperse a lot of invasive plants. However, in my study sites, it was pretty much dominantly native. The thimbleberry that was present was limited to the sides of trails and wasn't very common. So I think it has everything to do with the availability of the fruit that's there, and just it's easier for the birds to get the fruit that's readily available. So that would be reflective of what's in their diet. Especially at the Pu'u Maka'ala, the study site or the banding sites at Pu'u Maka'ala are pretty, I'm not sure if there's thimbleberry or blackberry. I can't remember, but if there is, it's really rare. So I think just fruit availability is influencing the diet composition. So it would actually be interesting to see if invaded forests or hybrid forests where ʻōmaʻo are present, if they are dispersing exotic fruits as well, just to see how adaptable they are to changes in the forest composition. But yeah, in general, that's what I think happened. So thank you.
Okay, any other questions?
I have a question. This is Pat. Let's see if I can get my... Hey, Koa. Great job. I just have a question. Do you feel like the exotic bird species are playing a greater role than the natives in dispersing any particular species of native fruits?
Yeah, definitely. I think just given the higher abundance of Huawei and Ridley, they're everywhere. If you go out in the forest, yeah, as you know, they're very common. They and just because of the higher numbers, I think that they definitely are a key disperser of fruits that they do consume, such as vaccinium and rubus, akala. So yeah, I agree.
Yeah. I was surprised about rubus. You didn't seem to get much rubus fruits in your, let's see, you didn't get it in your fecal samples from ʻōmaʻo, it seems like, but it was mostly in Leiothrix and Hawaiʻi. I was just really surprised about that.
Yeah, I wasn't sure about that either. I mean, just could have, my the time that I collected, I mean, rubus is fruits annually, right? So, or seasonally. So
yeah,
it could have just been like the timing of when the fecals were collected, and you know, there was variation in that. So that could have been an influence on why rubus wasn't represented well in the fecal of ʻōmaʻo. So
yeah, it seems to have a really narrow fruiting period.
Right, it's only in the summer for like 3 or 4 months or something. So
yeah, sure.
Yeah, in future it'd be better to evenly space out the nest netting and sampling to just get a better representation of diet throughout the year relative to biology.
Yeah, so I think the kava'u, the ilex, also seemed, which one was that, the ilex or the, yeah, it was most, oh no, ʻōmaʻo did ilex, yeah. Anyhow.
Okay, any further questions? Okay, well, if no more questions, let's congratulate Koa. Hey, congratulations, great job. We wish we could hug you and don you with leis right now, but we're doing that in spirit. And thank you so much for doing your presentation. Thank you all for coming. And if you can sign off now, we're going to continue to ask Koa a few more questions and talk about his next steps for his thesis. Thank you everyone so much for coming. We really appreciate it. And thank you, Koa, for sharing your work with us.
Yeah, mahalo everybody. Thank you for listening. Appreciate it.